Welcome to Decoding Healthcare Research, a podcast by Agora Project. Join us as we delve into the behind-the-scenes world of groundbreaking research and the dynamic healthcare industry, interviewing top paper authors, engaging experts on industry-related topics, and exploring medical subjects that affect our daily lives. And now, your host, Dr. E.F. Rain.
Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. Today, we will talk about the microbiome, diet in the context of cancer. So this is like three topics in one. And to discuss this interaction between the three, we will talk to our guest. She's Dr. Leigh Greathouse. She's Associate Professor of Nutrition Sciences at the Department of Human Services and Design at Baylor University. She's an expert in the topic, and it's an honor to have her here today with us. Welcome to our podcast, Dr. Greathouse.
Thanks for having me.
Thank you. So we will base our discussion on a paper that was published in the journal Neoplasia in 2022. The title of the paper is Diet Microbiome Interactions in Cancer Treatment, Opportunities and Challenges for Precision Nutrition in Cancer. So this paper called our attention because it touches on several aspects that are of critical importance when it comes to cancer treatment and how diet and the microbiome interact with each other. And that translates into response to treatment. And there are actually some insights about etiology and pathophysiology. So first of all, before we start digging into the paper, I want to ask Dr. Greathouse, what motivated you to explore this topic?
Sure. So I have to take a few steps back to tell you a little bit about my story. As a master's student in sports nutrition, I was diagnosed with a really rare form of cancer called uterine leiomyosarcoma. It was a stage 4. And so during that treatment time, I became very intrigued with how I could use nutrition to improve my overall status in response to treatment. And so from that time, I've been obsessed with trying to understand how we can use diet to improve treatment. Because for me, it made a tremendous difference, night and day difference for me when I changed my diet. And so during my time and efforts and doing my own research and having my own research lab, I become to understand that the microbiome and the diet are intimately linked together. And I believe that with a lot of the research coming out now, that idea is being driven home very much so, especially with regards to different treatment types for melanoma and glioblastoma, and hopefully in the future, other types of cancers. And I feel we are missing a big component of patient treatment because we don't yet have enough research to make nutrition a first-line treatment component along with other biologics and chemotherapeutics.
I see, and it has a lot of value, the fact that you kind of are on both sides of the equation, because you were a part of you experience being a patient, and at the same time that sparked your curiosity, and that definitely gives us an approach to what we are discussing here. And when we read about microbiome, diet, there are multiple publications in different contexts. But in the context of cancer specifically, nutrition is of critical importance. We know that the diet is directly linked in some aspects to the way you nourish yourself when you are undergoing treatment for cancer. So how do you reconcile, how do you put together diet microbiome in the context of cancer?
Sure. So currently we are just on the cusp of understanding how to utilize diet to change the microbiome into such a configuration that it's responsive to whatever therapy is being given to the patient. So right now, we really have a very rudimentary understanding of how the diet affects the microbiome. We understand at this point that you can make drastic changes in the microbiome structure and function in as little as 24 hours to 2 weeks. Because if you change your diet drastically enough, your microbiome will change fairly drastically. But again, the problem with implementing these types of studies is you have to have such a large cohort of people to study because the heterogeneity between the microbiome function and structure is so vast. You have to have a very large population. And then to even further drill down, it has to be, of course, by cancer type and by cancer treatment type even. So because we just have very little understanding at this point of how the diet truly affects the microbiome structure and function, and much less how the microbiome is affecting treatment response in individual people, it's a very large hill to climb at this point to get to precision nutrition medicine. But we are working towards understanding what kind and how much data it's going to take to get us there.
And this touches home with me because I am into data science, and I understand that the future is precision medicine and the application of data analysis to large amounts of data. And cancer and also the microbiome lend themselves to apply this. And when I read your paper, I think you are completely right. As precision medicine is something that is growing, precision nutrition has to go hand in hand. But there are many challenges that need to be overcome before that happens. But your paper starts touching on that. But before we go into that part, I want to clarify, or something that is important for our audience. What is the challenge, or what is the problem that malnutrition is imposing on the patients on cancer therapy? What is the consequence of that?
Sure. So there's several consequences, and I'll first state this. Even just the definition of malnutrition is a challenge because it is not well defined across medical disciplines, and that was come out in a NIH workshop where this was actually discussed. So one of our first challenges is in fact just defining the term malnutrition and what that means. The other problem with malnutrition in cancer patients is it doesn't allow the patient to fully respond to the treatment as well as they probably could, and leads to more severe side effects, toxicities, allowing or causing, dose reduction or stoppage of treatment, when if they were nourished appropriately in the beginning and followed along with the appropriate staff, they might have been able to continue or not dose reduce those treatments. So there's lots of complications on the back end when patient nutrition is not fully taken into consideration. And a big problem of this is lack of staff to do so, lack of understanding at the healthcare provider level, and of course, just lack of definitions of what malnutrition actually means and what to do about it.
Yeah, and another aspect that is important, and I see in my practice as an anesthesiologist, I deal with some patients who are actively receiving treatment for cancer, is that for them, even if they wanted to have a good nutrition and a good diet, the side effects of the therapy impedes the patients to receive a good nutrition, and that's something that is difficult to overcome, right? And that takes me to the next step. What is the role of the microbiome in that interaction between diet, nutrition, and cancer? Where can we put that into the equation?
So that's a great question. So that is really at the crossroads of what we're trying to understand right now. With regards to malnutrition, the way that we understand how the diet interacts with the microbiome, as far as I understand, has really only come from a lot of childhood pediatric studies and demonstrating how protein energy malnutrition is affecting or affected by the gut microbiome, and how restructuring that can overcome those undernutrition in children with protein energy malnutrition. So I'm sure something along the same lines exists in terms of cancer patients, maybe with patients with cancer cachexia as well. And so we need a lot more research to understand just basically what happens to the gut microbiome structure, function, metabolites along the continuum of treatment in cancer patients, especially ones that have malnutrition or cancer cachexia, to understand what is actually driving these changes. Is it the actual disease, of course, itself that's driving it? How much of the treatment is driving the changes in the microbiome? So once we understand and can separate out the treatment effects and the tumor effects on the microbiome, then we can start to go back and answer the questions, how much of the diet is changing the microbiome, and not only structure, but function. So there's several challenges for us to surmount at this point, as just basic understanding of nutrients and foods effect on the microbiome in healthy people and in people with cancer. And then separately from that, the treatment and the tumor effects or the cancer effects on the microbiome itself as well. Once we have addressed both of those very complicated experiments, studies, I think only then can we start to truly be able to integrate the diet with the microbiome to improve treatment response to whatever biologic or chemotherapeutic anti-cancer therapy the patient is on. So there's a huge set of challenges ahead of us, but as you know, I'm passionate to try to figure this out.
Yeah, and we are still learning, not only in the context of cancer, like in every other context, we are learning about the microbiome. And the more we learn, the more we know how much is still to be apprehended. So let's talk about the microbiome a little bit. When we talk about it, there are multiple like types of bacteria, almost impossible to memorize. So that makes it difficult. But let's say that there are microbes that have positive effects and some others that have a negative effect. And when it comes to cancer, especially when the patient is receiving treatment, we probably want to maximize the positive effects of some strains and minimize or mitigate the effect of the negative ones. So let's talk about the positive effects of the microbiome. How does it improve the immune function, the gut permeability, and the structure of the gut? What can you tell us about it?
There's a lot there to unpack. So I'll start with the gut permeability first. So that goes back again to more of the diet. So you have to have a sufficient amount of usually dietary fibers is one that I always go back to give an example of that. So different types of carbohydrates are broken down by, of course, our enzymes, but also by bacterial enzymes or microbial enzymes. And those are, of course, transformed into products like short chain fatty acids. And those usually tend to improve the gut barrier function. There's a lot of other factors I'm not going to mention, but a lot of that is due to your diet to improve the gut barrier function, to produce those short chain fatty acids that actually also regulate T cells and make T cells proliferate into T regulatory cells. And so some of the other functions that are important with the microbiome and the immune system, for example, we know from a couple of different studies from Jennifer Wargo's group and Rosemina Goldschmied, they first demonstrated that in melanoma patients, they could predict response to immunotherapy, they believed in the beginning, by just looking at diversity of the gut microbiome. And I can go back and talk about that a little bit more and why that is likely. But later on, they went on to find out that it wasn't just the diversity of the microbiome. In fact, it really wasn't that. It was, in fact, several species of microbes, including Rhinococciaceae, which seems to be a very strong beneficial bacteria, at least for immunotherapy response. And so they went into animal models and asked the question, well, if they took the fecal samples from responders or non-responders and fed a high dietary fiber to the animals, what would happen to the immune cells? And so what they found out was that the specific bacteria were metabolizing these dietary fibers into metabolites, specifically cyclic dinucleotides. And these cyclic dinucleotides were triggering the sting response, so that DNA damaging response, and then activating an interferon type 1 response. So the natural killer cells were being activated and anti-tumor macrophage were coming to play. So it was a byproduct from the microbes that were actually activating and recruiting these immune cells. And we've seen this happen in a handful of other studies as well, is that the presence of these microbes and potentially some of their metabolites is what is encouraging or activating the immune system, or encouraging the activation of CD8 T cells in the tumor microenvironment. Going back to diversity, I would say we know that for the most part, if I was to make a blanket statement of one thing we could probably do right now to improve the treatment response of most cancer patients, is to improve the diversity of their gut microbiome overall, because we know that having a very diverse food intake, mostly plants, fruits and vegetables, increases the diversity of the gut microbiome. And when you have a diverse gut microbiome, you have much more stability. And it's the stability that acts kind of like a buffer against insults from the environment or from things coming into the host system. And you also have multiple backup systems within your microbiome when you have much more diversity, because a lot of our microbes share the same metabolic pathways. But when your diversity is diminished significantly, those metabolic pathways are only relegated to a certain clade of bacteria now, and your ability to withstand insults in the gut or in your microbiome overall are greatly diminished. So I believe one of the things that we could do immediately for most cancer patients is just increase the diversity of their gut microbiome.
So when you talk about increasing diversity, how can you do that? Because when I see the formulations for probiotics, again, a lot of different types of bacteria, you don't even know which one is good. I don't even know if those ones are going to colonize. So should we focus more on fiber rather than probiotics?
In my opinion, at this point, I believe we should, because we don't have enough information to recommend the right type of probiotics. I just don't think they're out there yet. They haven't been really well developed or characterized. And usually what happens when you introduce a probiotic is it unnaturally, artificially inflates the diversity of one type of clade or species, genera of microbes, and then you actually lose diversity and you become reliant upon this one set of microbes. So really, truthfully, the only way currently that I know of to improve diversity is surely just increasing the amount of and types of food that we have. But going back to your original problem that you mentioned is, yeah, a lot of these patients just can't eat those types of foods at this point. So we have to be a lot more creative in identifying ways to improve the microbes that are most beneficial and not just the diversity for those patients who can't eat due to stomatitis or swallowing or just cachexia, things that are not allowing them to eat regularly. So I think we have to maybe take a different approach with those individuals and be much more specific with what types of foods that might encourage the microbes, or identifying later on which microbes we can introduce to encourage in treatment response.
Yeah, and I think that's also true outside of the realm of cancer for the same reasons that you are mentioning now. Let's move on to the effect, the side effects of the different therapies, and the effectiveness of the therapies, and how the microbiome can be a facilitator for those effects. You mentioned the boost of immunotherapy with microbiome. What is the role of microbiome when you are receiving chemotherapy or radiotherapy? Are they equally effective?
So that is less well understood because we just don't have enough, I would say, longitudinal data from cancer patients that are undergoing radio and chemotherapy. Most of these studies are extremely small, and it's really hard to understand exactly what's happening to these people. But I would say with the studies that we currently have, at least with some specific types of cancer, it does seem to make a difference as where you start off baseline. Meaning, like, if your gut microbiome is diverse and with certain types of beneficial microbes already colonized, then it appears that you seem to have a better response, again, because it goes back to that diversity problem. If you don't have a high diversity, which was usually indicated by a prudent or healthy diet, your ability to withstand some of the toxic side effects from these chemotherapies and radiotherapy is diminished. We know that some of these effects have to do with the toll-like receptor effects as well, and creating some of these toxicities. And so again, most of the studies, even though they're quite small, do seem to point to overall diversity as shoring up the system to reduce toxic side effects.
Yeah, and this is probably one of the most important things when it comes to cancer, to be able, as you said, to be creative and to provide the adequate nutrition and support to the microbiome, for that matter. So let's talk about dietary recommendations for someone who is entering this stage of treatment with different modalities. So we talked about probiotics. Probably there is some role for prebiotics as well.
Yeah. So I would probably say I probably would discourage any use of probiotics right before treatment. And the things that I've been telling physicians right now, and when they ask me this question, is you need to help the patient prep before you start in on chemotherapy or immunotherapy, meaning you need to prep their gut microbiome with as healthy as a diet you can get them to eat, whether that be, just improving the amount of different types of fruits and vegetables and whole grains that they are being exposed to. It has to be a drastic change, and that's what it takes. And about, I would say, at least 2 weeks on this drastically improved healthy diet with high fiber and fruits and vegetables, whole foods and vegetables, as healthy as they can do for just 2 weeks, I think would set them up for much better response to treatment, especially with immunotherapy and chemotherapy. Radiotherapy is still kind of nebulous at this point, but I believe with the current data that we have, that would be, I believe, the best option, because once you start in on the therapy, they're just going to go downhill with, side effects, nausea, vomiting, stomatitis, all of the side effects that reduce your ability to eat properly. So I think getting them, their microbiome beefed up before they go into therapy might give them a better chance to withstand the side effects.
Yeah, and that's actually great advice. And what are your thoughts about, in the context of inflammatory diseases, not necessarily cancer? There are several papers mentioning the benefits of different dietary strategies, such as ketogenic diets, different types of fasting mimicking diets. So, do you think there is some room for these innovative types of approach to nutrition?
Yeah, absolutely. I know you weren't really interested necessarily in the cancer approach, but I have seen a handful of studies looking at the fasting mimicking diet, which I think could be fantastic, given what I know it has to do with metabolism and reducing, growth factor expression or availability. It seems to improve dramatically the CD8 T cell activation as well. So I think the fasting mimicking diet would be a fantastic thing to do a lot more research into, especially for cancer patients. I don't know exactly what the effect on the microbiome is. I haven't seen a whole lot of research in that context with regards to cancer, but I am excited to see where that research goes. I will say that with the ketogenic diet, my colleague Jethro Hugh at Cedars-Sinai is doing a phase 2 clinical trial in glioblastoma with patients on the ketogenic diet. And that seems to be very beneficial, at least from the phase 1 study, for improving response to chemotherapy for glioblastoma. And we know that the gut microbiome is definitely affected by the ketogenic diet. It does seem to have some beneficial effects on the immune system as well. So I think, again, it's going to be really specific to the cancer type because of the metabolism of different types of cancers. Maybe some types of cancers are more addicted to the Warburg effect and growth factors, whereas some cancers like glioblastoma may benefit from starvation of glucose. So I think it really comes down to understanding metabolism of the tumor and the immune microenvironment in which it's living.
Yeah, and I understand that it's difficult to give recommendations that apply to everybody, and that takes us to the next point that I think is probably the most interesting in the paper is when you start talking about precision nutrition, and in that context, a term that actually caught my attention was molecular pathological epidemiology. What is that, and what do you think should be done in order to make the nutritional recommendations in cancer more individualized?
Yeah, so that actually came from a reviewer comment. They wanted me to add that in the paper. So actually that is some work that is being done out of Harvard. And they're doing a lot of molecular pathology of the tumors and looking at signatures. So I'll explain it in this way. So we've been talking about this increase in early onset colorectal cancer, and we're trying to look for molecular signatures as to give us some directions on what exposures might be driving this early onset colorectal cancer risk. And so we're looking at the molecular signatures of early onset colorectal cancer versus late onset. And so if we know the signatures of those mutations, we can then track that backwards and say, well, we know these exposures, dietary or chemical, are associated with these ADOXO-DG or C to T transformations or something like that. So when we say molecular pathology and epidemiology, that's what we're indicating with that term. And I believe we can do something similar with the microbiome. If we can say these signatures are associated with this type of microbiome, then we might be able to use that as biomarkers or risk factors and modify the biomarkers or the microbiome to change that signature.
I see, and it's a complex topic actually, but from my perspective, as someone who is not an expert, I see that the combination or the interaction between genetics, nutrition, and the microbiome is going to lead to this concept of precision nutrition. The what needs to happen for this concept to actually be operationalized and to be actually a reality?
Yeah, I'm my vision right now to operationalize precision nutrition, I think is going to be most efficiently done, and I hate using this term because it's used so much, is using different forms of artificial intelligence. So there is so much complicated data to wade through at this point. I don't think using our standard molecular epidemiology, molecular biology approaches are going to get us there quickly enough. It's really going to take more of a systems biology and AI approach to solve this problem. So the approach that I'm currently working on with my colleague Nick Chia at Argonne National Labs, he has a physicist and AI modeler, and what we're doing right now is we're working together to build simulations of the data that we currently have. So we are working with an AI model in which we train the model to utilize or categorize data that we already know into modules that we can use to train the AI algorithm, then to expand upon the knowledge we already have. And once we have that trained, we can then create simulation models of certain tumor mutations. And Nick has already actually done this. We have certain tumor mutation signatures that we know that are associated with different microbes. And once we have the microbes and we know what metabolism and outputs they have, then we can start to simulate those signatures in the tumor and the microbiome, and then ask what changes if we change the dial here on the microbiome, what tumor mutations might change as well, and vice versa, because there's an interaction between both. It's not just one way, right? And so once we understand how we change the microbiome, how that changes the tumor microenvironment, the mutations, then we can understand how to make it work together. And so once we understand that, then we can add the dietary component. And we understand how the diet now starts to affect the microbiome. And then once we understand how to change those dials, then we can change all three at the same time and understand how to make that work for individual cancer patients. But again, it's going to take a lot of data. And I think right now we are going to have to use the best simulation models that we can. And that will tell us essentially kind of how much real world data we'll need to train the model to actually understand how much variance is there in the system between cancer patients and how much can we have an effect on changing the microbiome to change the tumor metabolism in response.
Yes, and I am sure that as you are able to put all these data together and basically come up with actionable insights that can be translated into actual therapeutics, we are going to start finding more answers at the individual level. That's for sure. And before we come to a close, I want to ask you something. Now going to the practical side of all this. So as precision nutrition is going to emerge and be more used, do you think the healthcare systems are prepared for that, to deliver that type of care?
Not right now. They're going to have to rethink how they deliver care because obviously there's a shortage of doctors. I can absolutely tell you there's a shortage of healthcare providers overall, nurses, obviously dietitians, to do this type of work. And what I imagine is going to have to happen is you're going to have to rely on more types of either telemedicine or mobile health algorithms to walk the patient through what's going on with them right now to evaluate in order to make basic recommendations that hopefully are guided by current healthcare practices in the system to alleviate some of the burden off the physicians and other healthcare providers to make specific recommendations. So one of the things that I'm working on, I have a company called Cancer Warrior Health, and we are building a platform right now that integrates nutrition, mental and physical health recommendations all in one. And currently, we have a prototype platform. But what I would love to see happen as we expand this platform is to allow patients to track their own health, mentally, physically, nutritionally, so we can at least prevent them from dropping off the cliff into malnutrition or physical disabilities that might not necessarily have to happen had we been proactive, and especially mental health, because I think all of these three things are critically important. And without one, you can fall into despair in the other areas as well. And so I think we'll have to have a lot of data, again, to train these modules to really recognize, to be able to do precision nutrition, precision mental and physical health. But I think having these integrated would do a lot for these patients to at least be a stopgap for the healthcare profession. You know, if we could add this into Epic system on the backside and say, you know, physician, your patient now is red flagged as malnourished. They also have to be going down the hill mentally right now. These are things that you might want to pick up on and address at your next visit. We can also make referrals as well to healthcare professionals that can fill in those gaps while if they can't make it to the physician or the healthcare provider where they are, because maybe they're rural. So I think it's a being creative in that type of way to utilize our online platforms, our mobile health platforms to be that stopgap for the healthcare provider and the healthcare system overall. I think we're going to have to get really creative in that space.
Yeah, and I am excited with that, and I really anticipate that these decision support systems are going to become mainstream for sure. And actually, I would like to learn more about your company and the platform that you just mentioned. If you can share some details offline, so that we can also share with our audience what you are doing in that regard, that would be awesome. And this way, I think we can come to a close. But before, I want to thank you for sharing your wisdom with us, your knowledge, and this topic is particularly important. There are many patients going through difficult times, and the maximization of nutrition and enhancement of the microbiome as an ally in our fight against cancer is something that definitely is going to be of value for all of our audience. Thank you so much, Dr. Greathouse. Thank you for being with us.
Thanks for having me.
Thank you. And for our audience, you will find the link to the paper in the description, as well as information that Dr. Greathouse is going to share with us about all these platforms and the systems that she's working on. Don't forget to leave us feedback in the comment section, and see you in the next episode of Decoding Healthcare Research.
Thank you so much for tuning into this episode. If you enjoyed it, please don't forget to give us a thumbs up and share it with your friends and family. Make sure to subscribe to our channel and hit that notification bell so you never miss an episode. If you have any questions or thoughts about today's topic, we'd love to hear from you. Feel free to leave your comments down below. For more information and references related to today's discussion, you can find them in the video description below. We appreciate your support and look forward to having you back for our next episode.